Yeah, but we have this one right here.
Okay, I just need to...
Yeah. It'll work. It'll work. Can we quickly test this one? I don't want to... Unless you are okay with that. Okay, then let's use your computer.
Just for my purpose.
Yeah. Some people are like some people are going to plug in the computer, but like you want to have a minimal blocking the console. Pretty fast.
Yeah. Alright.
Yeah.
Everything updated.
Yeah, I have your file. Yeah.
So yeah.
My slides because I just finished them.
That's fine. You'll be able to plug it in.
Yeah, we're gonna.
Do you need anything?
Other one? This one's on. All right. Hello and welcome to the CCN 2024 Battle of the Metrics event. My name is Colin Conwell, and it's my pleasure to be your guide on this journey through the hot and sizzling, actively developing world, rapidly evolving world, an excellent universe of science that has flourished at the intersection of neuroscience and AI. Our event today, and the larger project that this event is part of, is motivated by one overarching question, which is, what do we want when we use models to map the brains, and how do we get it? And I'll clarify what that means in a few slides here. So the way that this event is going to work is we're going to have a whole bunch of people from across the universe of model-to-brain benchmarking methods speak about their opinions and their metrics that they prefer for a given kind of understanding that they want to extract from the brain. Now you'll notice I'm using particularly vague terms, but that's also because there's a meta question at play here, which is, what is it that we're trying to get as a community? What, with all the tools and the capabilities that we gained in the recent years of AI flourishing, have we actually come to learn and understand about the brain? And as a bit of background to this, I'll give you the origin story, at least from my perspective. So a few years ago, you'll note there's this wonderful thing that came out in the world called BrainScore. And when I first saw this, I thought, this is absolutely hands down the coolest thing that I've ever seen as a cognitive scientist, and I have to figure this out. And I loved the paradigm so much, in fact, that I went home and for almost 3 years straight, I tried to figure out how to do BrainScore myself. And at the end of that, what I arrived at was actually more questions than I had actually started out with when I began the endeavor of reverse engineering of BrainScore. And in particular, one particular thing that we noticed was this inability, it seemed, to differentiate models that seemingly should be very different. You'll notice that one of the main features of a platform like BrainScore is a leaderboard. You go onto the website, you click on a link, and you see a list of models that have performed better and worse in some set of benchmarks that have been defined by the team at BrainScore. And there's one very notable feature of this, which is that you'll note that the best and next best model in many of these benchmarks is very minimally different in terms of the scores that they achieve on that benchmark, but sometimes seemingly maximally different in terms of things like architecture, task, input, the kinds of computational principles that we would think we would be discovering by doing something like a model-to-brain benchmarking paradigm. And so this is something that we also noticed when we tried to reverse engineer BrainScore and sort of manipulate the models into a set of opportunistic experiments that we called them. And overall, we found very little difference in many of the fundamental features that you would think would characterize model difference, such as architecture, task, and the training diet on which they were trained. So this sort of kind of created, for me at least, a bit of an existential crisis because I had come so far only to feel that the tools I was developing were not necessarily giving me what I wanted, but I couldn't even articulate what it was that I did want. And this became particularly salient when I discovered this paper a few years ago by the excellent Alex Williams, another hero in the neuro-AI space. And this very quickly was something that I looked at and said, oh, I kind of have to understand this. I'll be fully disclosing. This had the triangle inequality, which, as somebody that came from comparative literature and the theater, I didn't even know what it was. But it seemed important, and these people were very smart. So I teamed up with Francisco Acosta, a student that was an early entrant to the Battle of the Metric contest, and he threw a bunch of these metrics at a task which was trying to understand cortical geometry. And despite the triangle inequality seeming very important from the mathematical perspective, it didn't actually seem to make much of a difference when it came to the questions that we had about neuroscience. And so I'll say, Battle of the Metrics, one thing I want to clarify, it's a catchy name, but there is not really a battle part to it. It's basically a bunch of people that have gotten together to try and understand this paradigm that we're developing a bit better. And also, very importantly, metrics here we're using as a very loose term. We use them actually in 3 kinds of ways. One of these ways is alignment metrics, the kinds of similarity scores that you might get between a model and a brain. Something like metametrics, which is something like the model differentiability that you see across many models taken in perspective together. And also auxiliary metrics, things like neural manifold statistics, and representational measures that you can do on individual models to try and understand what the structure of those representations are and the kinds of information gathering tasks that they may be subserving. And so with that I'll say, Battle of the Metrics is not actually Battle of the Metrics, but is a coming together of like-minded people, all equally excited by the prospect and wonders of AI, but equally passionate about building the right tools, techniques, and community necessary to make the kinds of progress we want to make on understanding the brain. whatever that actually means. And with that, I'll turn it over to our position speakers, who will be giving you a broad overview of the first part of the question: what is it that we want from our model-to-brain benchmarking paradigms?
Nope.
Oh, forgot to mention one important thing. On the website, which there was a link both on the CCN scheduled events page and elsewhere, you will find a link to a Q&A or Q&A form that you can fill out, and we will be actually synthesizing your questions, your feedback, your comments on specific talks, or general comments and feedback, using some help from generative AI, and hopefully facilitating a more active and more comprehensive Q&A. Please do comment, talk, and cheerio. Okay.
Oh, I have several microphones. I'm not sure what to do with them. I would like a few more. Thank you, Colin. All right. I will endeavor to actually add battles and jabs to this, unlike what Colin just said. OK, so comparing models and brains, what matters for similarity? Well, here we're going to start with the premise. We have a bunch of AI models that have learned to do something we care about. That is a precondition. I care about it. In this case, the thing that I care about that they do is they seem to have learned a useful representation of the world from visual image data that can support things like object categorization. We also have a biological visual system that we know has useful representations of the world because they're in our heads. And we know it supports many rich visual behaviors because we do them. And so the questions we have are these. How is patterned light that's actively sensed through the retina transformed into these useful representations? What's that flow of information processing to convert one format into the next, and what are these representational formats like? Can I get an intuition of how that's even possible? So these are open questions about the brain, and we can take lots of measurements to different stimuli, but those measurements don't give us direct access to answer these questions, right? They're the kinds of things we can use. We still got to figure out these are the questions. This is what I want. And the question is, can we use models to help me answer these questions? So I've defined them. And note, these questions are not like, what's the model that best predicts the brain? Like, this is a specific brain-focused question about these formats. So one way to start at this is go, well, can we see if the representations learned by these models are similar to those that we seem to be able to measure in the brain? And this really raises a question of how do we know if two representations are similar? And I had the privilege of getting to do this talk with Rosa Cow, who I think has really clear thoughts on this. For instance, this paper, Putting Representations to Use. And she makes several points. One is that similarity is fraught as a philosophical concept, and it's at best context-dependent. Here's an example. Which of these two are more similar? OK, do you see the problem? Baseball could go with bat, but baseball could go with orange. OK, so there's two different answers. How do we want to balance these? Is there a single similarity that's going to make sense of this kind of relationship? OK, and so her stance, which I share, is that DNN model, say DNN model layers, will have similar representations to brain areas if the resulting representations are used in the same way for the next stage of processing or for the behavior. I think that is right on, but how do you measure that? OK, so let's see what we can do. And I think important for understanding this is some terminological clarity we might need to articulate this need. And I've learned there's a nice GAC that's really gone after this. Rosa, part of it, and there was a poster earlier, which I think articulates it even more than I'm going to do here, but I'm going to lay out one critical distinction, which I hope we can all work from. And the second thing I'll do is highlight the implications this has for how we want to think about how we should use models to tell us about brains. OK, so the distinction is what I think of as representations with and without a capital R. So representation with a capital R means it's an encoding of information that has a functional role. you have a representational capacity, and it only exists when that content of that representation is functionally used for the thing it's for. So as an example of where this kind of rears its head is, when we're measuring from category selective regions, and we can, say, decode house information from FFA, does that mean that FFA is representing house information? Well, you might argue no, because the system doesn't use those responses to recognize houses. Maybe it does, maybe it doesn't. But if it doesn't, then that would be not representation with a capital R, right? It's not used, so don't call that a representation. And so this really makes you have to understand the readout commitments. There's a population of information, which we can decode lots of it, but how is the brain actually reading out from that? Or how is information actually propagating from that code to the next stage of processing? This is hard to know. And so, but one thing that's easy to know, or easier to know, is what I think of as representation with a lowercase r. And this is how I think, this is just measurement. This is the encoding format. So we can say a pixel-wise representation. OK, each pixel is a basis set, and images land in this space, and boom, that's what it is. Or a Gabor jet representation. A V1 representation takes the units you happen to measure from V1. Those are your axes. Great. This is just an encoding format. This is safe. This is just measurement. It's true in some measurement way if you took good measures. But it's a slippery slope when we measure these things and then say, thus we have representation with a capital R. Like, I think we're a little too casual going between the measurement and the commitment of whether it's actually used in the system. So comparing representations when the bases match, like pixel-wise, two images in the same size, and you can just subtract the pixels, right? That's easy. But when we have to compare across representational formats when the basis set doesn't match, now what? And there's been a number of linking methods that have been developed, but if you cast this in the context of lowercase and uppercase representation, then these are making implicit readout assumptions. For instance, say you have a model layer and some images in there, and then you have a brain area, and the same images and the basis set don't match. We can do something like classic RSA, thank you, Nico, and compute the pairwise distance between all of these things, and you don't have to have the basis match. This is the first way we started to figure out, what if there's not a one-to-one match? What do we do? This is a configuration match. But in the context of what this is implying about capital R representation, it's implying that tuning directions are not the relevant information-carrying signal because it's invariant to the different rotations. It's also implying that there's sort of a population-level commitment that the span of these things in this basis set is sort of matched in an important way to the span of the second set of things. And it's possibly a commitment to that the readout needs to have full access to all the neurons to compute the relevant distances. So another thing we might do is change the target. Maybe we do care about tuning, so let's make an encoding model predict the tuning of this voxel or this neuron. And here the standard approach is to do some sort of regression, regularized ridge. So what are these commitments? Here, we're saying the tuning is mattering for reading out of this neuron's role, and that's sort of implicit in the fact that I made the units tuning my target of interest. But by allowing reweighting of all the features, especially if it's unregularized, is sort of a implicit statement that we think that can be arbitrarily related to the tuning of the units in the deep neural network. So do we think tuning doesn't matter in models? And these are just standard practices as we're grappling with these tools, but I think they imply or they reveal that we lack deep agreement on what the information-carrying signals are in these systems. So how do we know what the information-carrying signal is? Rosa says, well, follow the operations of the next stage. And, that's a little tricky in brains because we don't have access to the connectome and it's loopy, and I don't quite know how to do it. I think it's the right thing to do, but I don't know how to do it. But you know what? We can do it in feed-forward models that are like hierarchical. We could OK, thanks. So maybe there's hope to sort of tackle this question in the models. And if we think tuning is the relevant thing, well, in a way, the next layer is just weighted combinations of the tuning of the previous layer. So I can kind of understand how information is going from one stage to the next, how it's flowing, how it's getting routed through the network. With an RDM, you've now changed it to reflect the distance between conditions. Is that number actually playing a functional role in the computations of the next layer? I think that's an important question or challenge or poke or jab for the RDM people. Like, I would like to know. Maybe it is, but I think we should ask. And so in general, if we want to go from representation, measurement, to representation, functional role, since it's hard to trace the path, another strategy is like, well, what if we get as close to a one-to-one match as possible? And this sort of brings us to different frameworks of how these AI models are related to brains. And I think some people in this room, and I think this is sort of the brain score side of things, want these models to be models of the neural system, where there's some direct correspondence, and you want to find the best model, and there are some good ones, and I think Martin Shrimp said, we don't need to use AlexNet anymore, ResNet beats it. Like, there's this sense that there's one that we want to find. And here the goal is to add biological detail and add more measures, and I think this cashes out ultimately in a one-to-one in silico brain replica if all neural measurements and biological detail are in play, which is interesting and valuable, but I think it's not the only way and the only way to get information from these models. Another way that I think that they are useful is to think of them as model organisms. We study Drosophila, we study mice, we study monkeys, and we think that we can learn something about how those systems work. And we don't expect it to be exactly the same, but we think we might get insights into how the human brain works given those. And so here, the link is we study the system because it has an interesting common behavior, maybe in a simpler setting. And we're going to try and look for emergent common principles. And so we do science on the models to get there. And this kind of work is safe, because we can there's an answer. We can go, does this mechanism that we add or take away give rise to this representational capacity? And we know representation with a capital R because we just define it and measure it, and we can see if it's in the model. Does the model have this behavior or not? And so this way, it's nice because it's safe and true. And it's another question whether that's how the brain does it, but at least we're making progress on how things might work, and that's a pretty good start. So if we take this model organism framework, maybe we should change the goal from find an exact match to the next stage to discover the principles of readout or the principles of transmission from one stage to the next. Some intuitions on this front. I think ReLUs are really interesting because they put some clear constraints on what can make it through. And it turns out you don't need, a unit up here doesn't need to have weight on all the other units. Like it only sort of has high weights on a few of them, and you can prune away lots of units and still preserve this other next stage unit's function. So we've been thinking of this as positive information signaling through these networks, and if that's how information passes through, then we try to make the case that it makes sense to do sparse positive constraints matching tuning directions. So you're trying to find, so you shouldn't be able to negatively weight a neuron, a deep neural network unit, because if that's before the ReLU, that information just doesn't make it through to the next stage. The DeepNet couldn't even use that negatively weighted neuron. So these are trying to reflect the information processing constraints. And so, so here I would advocate for the position where the tuning vectors are really what's mattering for the model, because I think that makes sense of how information flows through these. And our metrics should minimize the amount of reorientation or gymnastics that are required to make one match the other. And we've developed some metrics for that. Don't flip the tuning. In sum, I've tried to say we should be careful when we say representational similarity analysis. We mean like encoding similarity analysis, and that's quite different from functional role similarity. And maybe we need new terms because while representation with capital R is catchy, it's long and not so useful. So if anyone has any terms, let me know. Okay, and the other is if we can understand where in the inferential landscape we are, and it makes certain inferences safer. And I think thinking of these as model organisms where we're just trying to understand how they work can let us make real traction on how information flows, and then I think those can reveal principles, and that's what we want to get from our models to understand the brain. Thank you very much. Anytime.
Okay, so
Do you have presenter now? go, sorry.
Yeah, go ahead.
Is this the start of it?
Oh no, you have it all the way up.
Here we go.
There we go. Really quickly, just. And then you can press play. Oh, okay. That's so sorry. Let me quickly just do. Play. I'm just gonna expand. There we go.
Okay, thank you.
Thank you so much.
Okay, so we now have many different computational models, thanks to deep learning, many different models that we can study as theories of information processing in the brain. And there's been a proliferation of studies that have compared these deep neural networks to human brain representations. Now, often the goal of these studies is to find the right neural network to explain a brain system. That is usually to find the right kind of architecture, the right kind of objective function, the right kind of training data, the right ingredients that are needed to explain the information processing mechanisms in a brain region, and also what objective it might be optimized for. But a number of studies now have demonstrated that this research program faces a major challenge. And that is that there are many diverse kinds of neural networks that seem to be equally good models of human brain representations. So I'm focusing here on vision, but this is really a general trend in the field right now. So what these results are telling us is that it's becoming increasingly difficult to adjudicate between models that make very different kinds of theoretical commitments for what's needed to explain brain representations, right? These are commitments to things like functional architecture, objective function, training data, and so on. And so the question is, how do we move forward from here? How do we deal with all of these competing models? So here I'm showing a schematic from a recent Perspective article on the Neuroconnectionist research program. And I want to use this schematic to highlight some of the ideas that have been proposed for how we should move forward. Okay, so first of all, we're all in agreement that we'd like to have better data sets, especially larger and more complex data sets that can really challenge our models. But others have been proposing more creative ways of addressing this problem. So for example, it's been suggested that we need better experimental approaches that do targeted manipulations of models, pit models against one another, allow us to figure out what are the most important factors that explain the success of the best models. Right? And these factors are often things like architecture and objective function. Others have argued that we need better metrics for pulling apart models. Right? So Talia just talked about this idea. The key argument here is that maybe our metrics are too flexible. Right? They allow for too much flexibility when we're mapping high-dimensional neural networks to patterns of brain activity. And this flexibility is just making all of the models look equally good. And the argument is that if we can find more principled biases for our metrics, right, theory-driven biases, we can better differentiate which models are right and which models are wrong. And this is really the central theme of all of these proposals. The idea is to find better ways of pulling out the important differences in these models. So for the remainder of the talk, I'm going to argue for a different perspective on this problem. So I'm going to argue is going to start with a really basic question. And that is, what if these models aren't actually different from one another? What if at a fundamental level, these models are all effectively the same? If that were the case, then trying to pull out the differences among them wouldn't really be the best way forward. All right, to make this idea more concrete, I'm going to briefly tell you about a finding from my group. So this is a study in which we examined diverse neural network models of vision to determine the degree to which they rely on representations that are universal, that is, shared across networks. So specifically, we examined every latent dimension in a set of networks with different kinds of architectures. So I'm focusing on this analysis of architectures here, but we've done the same thing when comparing models that were trained with different objective functions, and the results that I'll show you today are representative of what we see in that case too. All right, so what we did is for every dimension in these networks, we computed a universality score, which measures the degree to which a dimension is shared across networks. So dimensions that have high universality scores are found in nearly every network that we examine, and dimensions that have low universality scores only emerge in specific kinds of network architectures. We also computed another metric for these dimensions called brain similarity. So here, we're measuring the similarity between a neural network dimension and the representations of the human brain. And we did this by looking at the fMRI responses in visual cortex from the natural scenes data set. So here are the results for computing these two metrics on 150,000 dimensions from diverse neural network models. So what we can see here, oh, and I just explain what you're looking at here. So this is a density plot of these 150,000 points. And the orange dots are the average values in quantile bins of the data. Now what we can see is a really striking relationship between the universality of these dimensions and their similarities to the human brain. Specifically, what we see is that the dimensions that are most strongly shared with the human brain are those that are universal across networks. And in contrast, the dimensions that are model-specific, the ones that only emerge in specific kinds of architectures, these have very little in common with the human brain. And I want to point out that there are no data points in the upper left corner of this plot. Right, so this is where we would find dimensions that only emerge under specific kinds of network architectures and that are also shared with the brain. Right, this is actually the exact kind of thing that we're looking for when we pit models against one another. And this analysis is suggesting that those kinds of things are rare. All right, so what these results are telling us is that the most strongly brain-aligned representations in neural networks are universal. They emerge in all networks despite major differences in their designs. And this suggests that we need a different way of thinking about the relationship between neural networks and the human brain. It seems that we can't understand this relationship in terms of shared architectures or shared objective functions. It seems like we need a much deeper explanation. So how are we going to get at this deeper explanation? So I'm going to say a little bit about what I think is the kind of approach that we need. But I first want to just take a step back and consider what is it that we're going after as a field in general? What kinds of scientific explanations do we want? And I think it's helpful to keep in mind that in other fields, many fundamental principles in nature are often independent of the specific characteristics of the systems that they apply to. So just to give a few examples of here, this is true for things like natural selection, thermodynamics, allometric scaling laws in biology. These are all theories that describe the behaviors of highly diverse systems without making reference to the detailed characteristics of those systems. And I think that the field of computational neuroscience is seeing some really compelling evidence that diverse networks are converging to common representations that are independent of the details of their designs. Instead of thinking about this as a problem that we need to solve by pulling these networks apart, I think we should be thinking about this as a key phenomenon that we want to explain. And that's because I think it points to some really fundamental principles of neural representation that we don't understand yet. Okay, so if you want to go after these universal representations and their underlying dimensions, what kind of approach is needed? I want to end by just drawing a contrast between the approach that I'm arguing for and what others have been suggesting recently. So first, it's been argued that we need better ways of pulling out the important differences among models. In contrast, what I'm arguing is that we need better ways of finding the core fundamental similarities between models. Our focus should be more on the similarities among these systems, not the differences. It's been argued that we need better metrics, less flexibility, so that we can better differentiate models. I think this flexibility is going to be crucial, actually. I think we need flexible methods to pull out the latent properties that are shared by diverse systems and systems that may appear to be highly distinct at a surface level. Okay, and I just want to say one more thing before I end here, which is that we don't know what these universal principles are yet, and we don't know exactly how we're going to discover them. But I think if we're going to have any hope of discovering these things, we're going to need theories and methods that go after these sort of common properties that emerge across diverse systems and are independent of the details of their designs. Thank you so much for your attention.
I think something fell.
Oh, yeah, yeah. Sorry. Okay. Okay, that's right here.
And there's a power cord?
Oh, yeah, yeah. Did you want the marker?
Yes.
Okay, sure.
Oh, great. Oh, this is the wrong thing. Oh, no, it's not. I mean, it's the wrong power cord.
That is not for yours?
Well, it's certainly not the, yeah, this is a really old one. All right. I think I'm okay though.
You're right, you're right.
Yeah. Okay. Okay, so this is my title, and this is the subtitle, I guess. Okay, so what I want to talk about is meta principles for metric selection, and of course, the obvious in many situations there is uncertainty about which metric to use. There's no obvious ground truth to know whether the answer by the metric is correct. So how do we deal with this? There's many possible metrics, but I think there are also some universal principles, and I sort of, I don't know, tried to enumerate what I thought they were, and I'd like to talk about them. But before that, I don't think this is exactly true, but I'm going to imagine for most purposes that every metric has an implied transform class behind it. So it can be very loose, such as nonlinear transforms. It can be somewhat loose, such as linear prediction, so like the general linear group. It can be a bit stricter, like RSA, which isn't exactly the orthogonal group, but it's kind of. And then it can be strict, such as like one-to-one matching, so basically permutation group. Okay. Given that, these are the principles I'm going to talk about, if I can get through them. One is using obvious ground truth when you can. So let's start with that. A phenomenon is obviously ground truthy, that was the word I came up for it, if its existence can be visualized and certified by a metric that's really obviously correct, and nobody doubts about it, and it is usually, like, indeed close to universally observed. So like this. Okay? It's not quite right because, of course, there are, like, you know, it's like general relativity in QFT, that's more right, but it's still pretty good. And everybody agrees that, you know, the way we normally think of mechanics is correct. So let's talk about ground truthiness for a second. This is an image of the type of mapping people would like to do in the brain score world. One key feature of the ventral visual pathway in macaques, maybe a little less clear in humans, is there's a hierarchical ordering. It would be great if there was a metric of this kind so we could be measuring it. But hierarchy in macaques is about as strong a ground truthy statement as anything in systems neuro. So you really like to have intermodel distances from whatever your metric is that replicate the hierarchy. So, you know, the thing on the upper right, that's the best. Okay? If you figure out what the distances are for various metric types and then reduce them to RSA, you know, put a 2D RDM, actually you find that obviously ground truthy phenomenon, some metrics seem to find it, especially the looser ones, and capture it better than strict metrics. Okay? So here's a principle. Your metric should capture the obviously ground truthy statements when they can be found. Okay? Next, systems identifiability. So I'm going to define systems is identified basically if over the instances of the system present in the real world, different individuals, okay, pairs of parts drawn from the instances across the population that are supposed to be the same are considered the same by your metric, and instances that are supposed to be different are different. So here's a principle. A metric should perform systems identification for any ground truthy neuroanatomical phenomena. You should see the structure emerge in the metric. So here's this is for various specific metrics. you can see that the hierarchy emerges better for some than others. And it's kind of interesting that counterintuitively, the stricter metrics are less effective than some looser ones at systems identifiability, because it means that you must need a metric that depends on not just separation between different parts, separation between different things, but also similarity between the same parts. And Miran will talk about how to make this measurement and explain the theory why this fact occurs. Okay, next one. I'm going to call this check then balance. Okay, a metric is robust to target sampling if you can choose whatever you want in the target. Linear regressions like that, doesn't matter how many units you pick in the target, the same result will come up with it. And it's desirable because oftentimes brains are very incompletely sampled. Coverage is what fraction is covered in the target in the sample you have. High coverage is obviously desirable. Symmetry is what we all know it to be. It's one of the things you need to have a true metric distance. And finally, symmetrization. You can take any metric and you can symmetrize it by averaging both directions. And it's desirable because it constructs a symmetric metric from a potentially non-symmetric one. Okay, so let's do an example. The thing on the top is target sampling robustness. The results are not confounded by sparse sampling. Then there's symmetry, is going both directions on this arrow. Linear mapping is a target sample robust metric, but it is not symmetric. The forward direction is L, going from model to brain, and the reverse direction is going from brain to model. RSA and 1-1 sampling are symmetric, but are not target sample robust. Here, it's almost the right principle that what you want to basically do is have both symmetry and target sampling robustness. Okay? It actually turns out that no symmetric metrics can be target sample robust. And I have a proof of that if you want to hear. But forward, okay, that thing going up is forward linear mapping, and the thing going down is reverse. The purpose of forward linear mapping is to determine if the model contains the brain, and the purpose of the reverse is to determine if the brain contains the model. The problem of reverse mode, because it is not target sampling robust when the coverage is low. And symmetricizing will not help. So there's two bad procedures. Use only a symmetric metric alone, and use only a target sampled robust metric and symmetrize. This is what we should be doing instead. I'm calling this check then balance. Take a target sampling robust metric, compute the forward direction, compute the reverse direction for multiple sizes of data subsamples. Extrapolate where the reverse size series, where the asymptote of that reverse prediction would be. If the extrapolated asymptote is statistically valid, go to the next step. Else stop, collect more data, and repeat. And then, if you can go forward, then you symmetrize the results. So that my principle is use check then balance, not either of those two bad procedures. Okay, so I'm going to now talk about self-supervised versus unsupervised models as a way to think about how problems are arising. So here's a result from my former student, my PhD student, Cheng-Shu Zhang. And what he found was, okay, yeah, this is a nice, quantitatively accurate, unsupervised model of a higher brain area. Self-supervised is as good from this data, but not really better than the supervised metric, at least not according to the forward linear mapping metric. But there are topographical phenomena in, say, the primate ventral visual pathway that are kind of obviously ground truthy. Like you can see pinwheels in macaque V1. Macaque are human, right? So because of that, among things, a couple of us teamed up to build a model, specifically called the TDAN, that was hoping to capture some of these topographical phenomena. It's just a picture of the model. And it kind of does a decent job on some of the phenomenology. The thing on the left is what the model thinks of in V1, and the thing on the right is what it thinks of some part of VTC. So there's a correlation here between many different metrics, both quantitatively across a wide variety of topographical phenomena. Okay? So on the other hand, we found that topographical phenomena don't much affect the features of the learning model, because this is linear regression. The yellow and the purple lines are pretty much right on top of each other. So there's really no differentiation. However, if you use one-to-one mapping of that strict kind, you actually see it. And indeed, it improves the predictivity of all of the, for all of the metrics, one-to-one, almost up to the human ceiling. So we actually see the unsupervised model as being substantially better under that kind of metric. So who's right? Forward linear mapping or one-to-one mapping? Well, there's two principles that can give us some indication. One, if many metrics converge on the same model rank order, the ranking is more likely to be correct. Okay? And as it happens for this domain, the obviously ground truthy principle also applies, which is why we did that. in some sense. But there's another idea that you might invoke here, which is refinement. Like, because the loose model shows, just the supervised and unsupervised about equal, and the stricter metric shows the two models as separate, maybe it's a refinement process. So, okay, this reminds me of celestial navigation. There's intergalactic navigation, and you don't really care which solar system you're pointing at in the galaxy you want to go to. In fact, you can't even see it. So as you get closer, you change your metric and your navigation system, and you go to the right galaxy, the right solar system, excuse me. And then, of course, as you learn more and more, you have to figure out how to do interplanetary navigation. Unfortunately, going from the biggest to the medium to the small is the hardest way to get somewhere, but so it is. So I think of when we went from pretty old school models that weren't super, but still had some real serious work done to make them, inventing confidence essentially. And then when that was kind of like doing intergalactic navigation. And then we got a little closer, and we had basically used the categorization performance metric, and we were able to get a bit better. So we sort of did interstellar navigation. But then when you're done with that, you have to do interplanetary navigation, and who knows what the model will look like? That's the big question marks, and who knows what the principle is like? That's what we're talking about today. For example, maybe some other task that is not really done by any model today. That's only one thing. Okay, I'm going to say two things that look complicated quickly. So imagine you have two distributions of individuals. This is this more sophisticated universal principle that can never be computed. So it's basically saying for any metric of any kind, all the metrics, any function, in fact, that you can figure out, you want the distribution over population replacements to have the same distributional value. So it's basically created by evaluating the metric, whatever it is, not even a metric, it's an M, a function, via sampling in each slot from say, the model population or the animal population. Okay? A k equals 1 version might be like a number of units selected for a given category. K equals 2 examples might include linear regression and one-to-one match or anything. It's matching populations. And the principle is distributional substitutability completely subsumes all metrics into a single idea. But it's totally infeasible to evaluate because you can't quantify over all functions. Okay, at least not in a very direct way. So I'm finally going to end with this last thing, which is imagine, okay, we go back to that notion of the transform, okay? Now, put models and brains into this population together. The basic idea is that you should use the transform between brains, to get the empirical transform class, and then perform that check and balance procedure I told you about before with that same transform class on the population, including both the models and the brains. And this is really related to Talia's idea that readout representational mechanisms should reflect the biological representational mechanisms like the linear nonlinear Poisson neuron model. So it's basically saying the thing you use for your map should look like what you think the structure is doing. And for me, that's very close to matching the actual transform class between the animals. So the principle is EDAT, that's the name for it, subsumes all the principles mentioned today, but is much more tractable to compute than the distributional substitutability idea. So those are the principles, and I think there's some chance that we could use something like the last one to make progress. Okay, thanks.
Thanks so much. Okay, so we're now moving towards the second portion of the talk, which is called the metric blitz, and we'll be trying to keep it very quick.
Yeah.
So please do take into consideration that we will likely not have a lot of time for Q&A. So definitely fill out the form online. Is anybody, people find that online? Yes? Okay, good. We did not comment. Okay, so make ample usage of that. We'll have a brief summary at the end. We're going to try to get through all the material that we have. All right.
Great. Okay, my title is Metrics and Experiments for Meaningful and Powerful Model Comparison. And Colin asked me to do 4 minutes on meaningful and 4 minutes on powerful, but unfortunately they'll be all intermingled, so you have to figure out which is which. So in representational similarity analysis, as we all know, we look at the representational geometry of a population code. and we characterize it by its RDM. So we have to choose an RDM estimator and an RDM comparator to define our metric or our measure more generally of representational similarity. And there's sort of an area that's kind of our comfort zone of well-understood representational similarity measures. And these are symmetric measures, so they're RDM comparators, and we're very familiar with them in this community. So it's the cosine similarity, the Pearson correlation, rank correlations, and then more recently, whitened RDM error measures, which generalize the linear CKA, which is very popular in machine learning, and also applicable to model-brain comparisons. And this is a measure that was discovered by Jørn Dietrichsen's lab. I think Jørn is here. And that's an extremely important addition to this collection of choices. While all of these are similarities, we can also derive dissimilarities that correspond to these similarities and that have the same implicit equivalence classes of representations. And in particular, we can easily derive RDM metrics by normalizing the RDMs and then measuring either the Euclidean distance or the Mahalanobis distance. And for different normalizations of the RDMs, these correspond one-to-one to these measures on the left here. And then there are also a couple of other RDM metrics that are popular in the literature, like the angle between the vectorized RDMs, or the candle tau A rank correlation distance, or the angular CKA, and all of these are RDM metrics. So while this may be a little bit confusing, we actually have a good understanding of how to choose a combination of RDM estimator and RDM comparator for a particular experiment. And it depends on the answers to 3 questions. The first question is, do the models make ratio scale dissimilarity predictions? If so, we should use a ratio scale comparator, and otherwise a rank comparator or a topology-based comparator. The second question is whether we've repeated each of our stimuli to get multiple pattern estimates for each stimulus. And the third question is whether the errors of the pattern estimates are independent within the partitions of our data set. If the answers to the last 2 questions are both no, then we have to change our experimental design. But if you answer at least one of these last 2 questions with yes, then there is a good solution. And so for each of these cases, we have a combination of RDM estimator and comparator that is clearly preferable to other choices for the analysis. So all of this is pretty straightforward. We can just use this diagram to choose. So that's very nice. But of course, there's a bigger picture, right? And I love this framework of generalized shape metrics that Alex Williams talked about yesterday, because it gives us, not because it gives us metrics, as Alex likes to stress. We have lots of metrics. That's not an issue. But I love it because it gives us a rigorous theoretical and mathematical framework for thinking about the bigger picture, for making those equivalence classes explicit. And this framework includes geometry-based and linear encoding style comparisons as special cases, but it also includes these more restrictive measures and as well as less restrictive measures. And you've already heard about some of these more restrictive measures as introduced in exciting work by Meenakshi Khosla and also Jacob Prince. These papers were already put up by previous speakers, by Talia, I think. And then among the less restrictive measures, where we're even more flexible than with geometry-based comparison and linear invariance, is topology-based RSA, which Baihan Lin, a PhD student who recently graduated, introduced in a paper that's out as a preprint and is also in press. So just briefly, here's an illustration of this for 4 different representations. So you have this one-dimensional representation in four different hypothetical brain representations. When we look at the representational dissimilarity matrices, these reflect the similarity between the RDMs, reflect the geometry of these different representations. but we can take the RDM and convert it into a weighted graph, and then we can measure the on-manifold representational geodesics. And when we look at these distances measured on the neural manifold, then we see that these two geometries there on the left, which are more topologically similar, actually appear more similar in these geodesic distance matrices. All of these measures of representational similarity work in the model comparative framework that Heiko Schütt, who's in the audience, has recently published. And it's implemented in our RSA toolbox. So you get these familiar bar graphs with noise ceilings and Golan wings that show you, which pairs of models are significantly different, and that's a very important aspect of this. And then we also have a new visualization, the model map, where at the center of which we have a black dot, which is the data RDM, and then the noise ceiling becomes a noise halo, and all the models are arranged in this map with better models closer to the data and worse models further away. And all the distances to the center are exactly right, but then the angles are chosen so as to tell us something about the relationship between the different model predictions. So models that predict more similar geometries will be placed closer together in this kind of map. A major challenge with all this, however, is that distinct models often predict biological representations of natural stimuli equally well. And there are many examples in the literature. Mick has given a deliciously provocative talk about this with a very different perspective from the one that I'm going to present, but I'm very interested in thinking more about Mick's arguments as well. Our answer is to design experiments to adjudicate between models that implement different theories. And what we can do in vision, for example, is make controversial sets of stimuli that are optimized computationally to make models disagree in terms of their predictions of brain representations or behavioral outputs like classifications. In this case, these are images where the two adversarially trained models here are very confident that they're looking at a Weimaraner dog, whereas the Inception and ResNet model, they're both very confident that they're looking at Persian cats in these images. Right? So these images can kind of reveal the shortcomings of models. They give us a very powerful hammer to reject models, and also a lot of statistical power in comparing models. So this framework is evolving, and Tao Golan and Wen Guo in the lab have been hard at work at this, and it's really coming together very nicely for images and faces, and also language, sentences, and for behavioral measures as well as brain activity. And there's also many other labs that are using sort of optimized sets of stimuli increasingly in the community now, like in this example from Egbal and F. Fedorenko. So artificial stimuli are attractive for giving us powerful model comparisons, and natural stimuli are attractive for being more meaningful because they engage higher level representations in the brain, and they test models in a domain that we care more about, where we want the models to explain the computations that brains perform. But in between these two, there is a continuum, and controversial stimuli allow us to navigate that continuum very flexibly, and therefore we think they're critical for making progress. So they provide a kind of synthesis of artificial and natural, these two kind of opposing approaches in the literature. So in conclusion, we want our experiments and metrics to be both meaningful and powerful. So we want the experiments to be ecologically relevant in the choice of stimuli and designed to adjudicate among interesting models. And we want them to be powerful, and we can make them powerful by optimizing the stimuli and making controversial stimuli that the models really disagree over. And our metrics should make our analysis meaningful by being invariant to the biological variation our models are not meant to capture, and they should be sensitive to computational signatures of alternative models. So the final point that I want to make is that the question of metrics is not merely a technical question. It's a question that poses a fundamental challenge for our community to define at what level of abstraction our models should explain brain computation. And so I think there will not be sort of a simple answer to this, but there will be many metrics, and we need to keep exploring them. And this is the way that we find out what the right level of abstraction is to understand how brains compute. Thank you very much.
Okay, we're good. Okay, so hi everyone. So today I am actually going to be talking about two projects. So I'm going to start off with talking about my work on model metamers and how they help reveal divergent invariances between biological and artificial neural networks. This was done during my PhD at MIT with Josh McDermott. And then I'm going to switch gears for the second half of this talk. My collaborator Abdul could not be here today. And so then I will talk about this other measure of brain model comparison, which is or trying to interpret one measure of brain model comparison using our spectral theory of neural prediction and alignment. Okay, so let's just jump in. So this is an example of using actually behavioral measures as a way to compare computational models with biological systems, because sometimes, especially when we're studying humans, one of our most powerful tools is actually behavior. And so if we can use some sort of a behavior to actually place constraints on what the underlying representations of a system actually are, that can be very powerful for comparing our models. So within this work, we were really interested in looking at whether the invariances that are learned by deep neural networks actually match up with the invariances of the human perceptual system. And so to do this, we're going to use the idea of metamers, which are two stimuli that are physically different, but that produce the same responses within a system. Okay, so this framework for model metamers basically takes, in this case, a neural network that's trained to do some tasks, such as word recognition or image classification, and then we're going to design synthetic stimuli that produce the same response within this computational model. So to get this stimulus, we'll just take our reference, feed it into the model, get the model activations, and then to generate a new synthetic stimulus, in this case a model metamer, we're going to start with a noise stimulus, and we'll just use gradient descent-based optimization to minimize the distance between the model activation for the original and the synthetic stimulus. And eventually, once we take enough steps in the correct direction, we will modify this input noise so that we get a new stimulus that produces nearly the same activations at some layer of our artificial neural network. And also, because this is a bottleneck layer of a network that is able to do a classification task, this network will also produce the same behavioral response for the stimuli. Okay, so this is how we're going to generate these stimuli. And so basically what we do is we run this type of optimization for many different layers of the network, and we generate these stimuli at many different stages. And what this means is we can actually see where the invariances arise along the hierarchical model. Because for the two stimuli that are generated, or for our natural stimulus and the stimulus that is generated from the particular stage of the model activations, once again, are the same for those two stimuli. And so the model is going to be invariant to any differences that are present between those two stimuli. Okay, so the model doesn't know the difference between these stimuli, and so what we'll do is then we'll take these metamers and we run a behavioral test with human observers where they see or hear a model metamer or a natural stimulus, and they just have to report the class that it belongs to. Okay, so a very quick summary of this work is that we found that these late stages of our standard neural network models have model metamers that are basically just unrecognizable to human observers. And so basically you can see this for the example where the metamers for the final stages looks like some sort of strange noise stimulus. And so this really reveals that the invariances of these models are not aligning with human perceptual invariances. But we actually found that there's some cases where you do see very clear differences between these models. For instance, these are metamers from what we'll call an adversarially trained network, and these are actually much more recognizable to human observers. Now, there's a lot in this paper to check out, but for this workshop, you might be asking, okay, well, how does this measure, this behavioral measure, compare to other metrics of brain model similarity? And so we actually showed that the metamer recognition dissociates from tests of adversarial robustness, shape versus texture bias, and other tests of out-of-distribution stimuli. So it seems like these types of model metamers are teaching us something fundamentally different about our model. But I want to zoom in on a comparison that we did between human recognition of the model metamers, which here is on the y-axis, with the IT prediction score from a regression-based measure on the x-axis. And this is sort of pointing out, in terms of these regression measures, something that a lot of people have been highlighting, which is that these regression measures are really not showing us differences between the models. So the improvement of human recognition on the model metamers didn't really lead us to improve neural predictions, in this case in IT. So how exactly can models with very different representations lead to similar regression-based prediction scores? And so that actually leads me into the second project that I want to talk about, which we refer to this project as SNAP, which is a spectral theory of neural alignment and prediction. And the word spectral here is going to refer to the fact that we're going to rely on the eigenvalues and eigenvectors of our models. And so basically, this is a theoretical analysis that investigates how the spectral properties of a computational model actually contribute to the fidelity of the brain predictions from that model. So this is just looking at this very common setup of building a regression model, but a regression map between the model activations and the neural responses. And what we're going to do, or we did in this paper, is we adapted a recent theory from deep network learning literature that was based on statistical physics. And this theory relates the model eigenvalues and eigenvectors to the error that's obtained from the regression mapping. And so here, just to get everyone on the same page, here we're talking about the error from regression, and you might be used to thinking about like an R-squared value or a correlation. And so here, whenever I say a better model, that means that there is lower error. Okay, and so the main point here is that we're going to use this recent theory to dig further into the regression analysis and essentially try and interpret why models are achieving the predictions that they do. So in 3 minutes, I can't get into the math, but the basic idea is that the eigenvalues of the model and the alignment between the eigenvectors of the model and the neural data will give us what we're going to call error modes. After defining these error modes, we're going to summarize them using geometric measures. In this case, what I'll call the error mode radius and the error mode dimension. And actually, the product of these two terms is the total neural prediction error. And so on this plot, if we draw contour lines, any point along this contour will have the same neural prediction error, even though they might have different error mode geometries. And so models can lie on different places on this plot. For instance, this model has a higher error mode dimension, so basically the error is more spherical. And then this lower model has a lower error mode dimension, which means that the error is basically concentrated on a small number of modes. So visualizing it in this way, we can see that these models have the same neural prediction error, but are achieving it in very different ways. And in fact, these example models are basically the trend that we see if we look at our standard models, which are here in blue, and our adversarially trained models, which are here in green. So each point here is a regression model from a single model layer, and errors are basically connecting the same layer from the model. So the standard model has error that's relatively evenly distributed, while our adversarially trained models have low error in some dimensions and high error in other dimensions. And this really helps explain how we saw that the metamer test revealed clear differences between the models, even though the neural predictions did not. So essentially, they're achieving these neural predictions in very different ways. And potentially, this also suggests ways we could improve our models. So just to sort of conclude, I also am somewhat conflict averse because I think that many of our methods of brain model similarity are quite interesting and useful. They each teach us different things, and understanding and interpreting each measure can actually help us with model development. So thanks again, and thanks to the organizers.
Hi, everyone. I'm Brian, and this is work done jointly with Jacob, Tongzo, and Philip. So much of my work focuses on how intelligence works, how human intelligence works, and also how machine intelligence works. And something that we've all observed is that intelligence at the representational level looks pretty similar between our AI models and our biological brains. If you record a human brain viewing a set of images and show that same set of images to a neural network, you find the activations of the patterns of activation are, oh, okay.
You want to mirror the machine. There you go.
Oh.
Your notes.
Yeah.
Okay, hang on. Technical difficulties. Screen mirroring. Use a separate display. The notes are covering the slides. There we go. Yep.
Should I? Do you want me
Yeah,
to
go
do
for it.
All right, everyone. Almost. We're almost ready. There we go. Oh, God. Yeah, let's just go full screen. I'm gonna exit. I'm gonna close that real fast, okay? I'm gonna close it and open it again. Okay, we're gonna play with the presenter notes.
Yep.
So that is, where was that one? Play full in full.
Oh, great. Sorry about that. So, oh, actually, the notes aren't showing up. Wait. The notes aren't showing up in that case. Oh, there are no notes on this.
Okay, 1 second. Play, where was the one with this?
I can use my own laptop.
No.
Okay.
Hot takes while we fix the technical difficulties are welcome.
Take that, you can use this side. Okay. Do you want it?
Consider that part of the
Yeah, I don't know what's happening.
of learning about
The presenter notes are not showing up. No, it should.
there's a lot of game, and there is progress to be made
Oh, there they are.
technological view of how we do that. So, keeping in mind,
This works?
This works.
Perfect.
simultaneously develop tools and infrastructure that help us advance in tech theory at speed is a fundamental way that we can keep things moving.
Thank you. Thanks, everyone. Okay, let's start again. Hi, everyone. I'm Brian, and this work is done jointly with Jacob, Tongzo, and Philip. So much of my research focuses on how intelligence works, how human intelligence works, and also how machine intelligence works. And something that we've observed is that intelligence at the representational level looks pretty similar between our AI models and our biological brains. If you record a human brain viewing a set of images and show that same set of images to a neural network, you find the patterns of activation are strikingly similar. I'll explain what these matrix patterns mean later, but just from visual inspection, they look pretty similar. It turns out it's not just one particular neural network architecture. It's not just ResNet. It's not just vision transformers. In fact, it seems like any competent vision model learns similar presentations to the brain. What gives? We were trying to figure out how intelligence works, and everything seems to work. So this led us to ask the following question. Do different models actually represent the world in different ways, or are they all somehow becoming alike? What does it mean for two representations to be alike? We'll restrict our attention to things that are vector embeddings, so mappings from something like data, like images, to a vector. And we're going to characterize this representation only in terms of its inner product kernel. This tells us how representation measures distance between different data points. So the kernel of a vision system is evaluated over this set of images, will generate a matrix. The kernel is saying, my representation of this orange is similar to my representation of this apple. And my representation of this elephant is different from this orange. Now we'll build a kernel matrix for vision and another corresponding kernel matrix for text. And if the kernel matrices are similar, as is the case here, the vision model and the text model are considered to be highly aligned. And this is what the community defines as representational alignment, the similarity of kernels, or at a high level, the similarity of similarities. We're ready to go over one of the main experiments in our paper. We measured the alignment between a set of pure vision models trained only on images and a set of pure language models trained only on text. Could these different models trained on different modalities end up learning the same kernel? Let's start with one hypothesis. Language is distinct from vision, and vision is distinct from language. Models are simply learning to specialize in the aspects of the modality they're trained on. Then there's the more optimistic perspective. Language models align more with vision models as language models become more capable. Better language models are also better vision models. Here's what we actually see in our results. Alignment increases as a function of language model performance. We test models of many different ranges of capabilities, from the 560 million parameter Bloom model to the 65 billion parameter Llama models. And note, this is alignment over Dino v2, a self-supervised model trained only on images without any semantic information. But this is just on Dino small. What about larger, more capable models? The alignment is even higher. You can see the alignment simply shifts upward with increasing vision model capability. So why is this happening? One explanation could be we're all training on the same data. We've all decided that the internet is the best data set for our models. But it can't be a superficial aspect of the data. I just showed you language models trained on language data and models trained on image data both arrive at similar kernels. Another explanation is that maybe we're all using the same neural network architecture. Maybe we're using transformers, right? But this isn't quite true. We tested convergence on common architectures as well and found the same kinds of trends. Commons learn similar kernels to transformers. So what's left? What's common between all these models? Our answer, it's the world. So here's our hypothesis. The picture that I have in mind is Plato's cave. Plato imagined these prisoners in a cave whose only experience of the world is shadows on the cave walls, but they somehow infer that there is a world out there. And it's an allegory because Plato is saying this is actually how it is in our regular lives. We can only access our observations. We can only infer that there is an ideal reality, a reality behind the scenes that creates those observations. So the picture we have in mind is that there's a world out there. For example, you could view this world through a camera, or you could view this world through text. If we learn representations from this world, these representations should become alike. And it's because they're generated by the same causal process and the same underlying reality. So our hypothesis is that as these representations should become alike. That's because they're generated by the same causal process. So different neural networks are converging to the same way of representing the world, and specifically to the same Platonic kernel. Thank you.
Okay, that works. Hi, everyone. I'm Abdul, and I'm going to tell you about the work that I've been doing with my collaborators here, Nagaz Zaslavsky and Colton Castle, Colin Conwell, and my advisor, Ev Fedorenko, on convergence between artificial and biological neural networks. So when we compare representation between brains and ANNs, say in visual domain, a common finding is that ANNs can predict patterns of fMRI activity to a similar degree. Here I'm just showing a toy scenario. Now the question is, can we say that all three of these ANN are equally good models of the brain region we're interested in? Inspired by work of Tal Golan, Nico Kriegeskorte, and others on controversial stimuli, we need to consider whether the stimuli can stress test the ANNs and elicit different representation between them when we compare them to the brain. Additionally, we want to stay within the domain of natural stimuli. In the case of language, we had to do that, and for vision, for similar reasons. So let's say we find stimuli that are represented differently across models. We can have two possible scenarios. In the first scenario, one of these ANNs is a better model of the region that we're interested in, and under these stress testing conditions, it would come out as a winner. We call this a representation individuality hypothesis, that between these three models, they do have meaningfully distinct representation, and only one of these representations align with the human brain. And in the second scenario, all of these ANNs are good models, and when we try to push their representations apart from each other, we also pushing them away from the representation in the brain. So we call this hypothesis universality or convergence, and in this story, there can be many models that by virtue of being trained on data with similar statistics arrive at a similar representational space, and this space is similar to that of humans. And we can check for these two scenarios and see which one is closer to reality. So we can define a measure to quantify agreement across models based on the representational similarity that you noticed in the prior presentations, and then do a sampling to put models in either high disagreement or in high agreement. So on natural scene data set, which has 1,000 images, we can find a random set of 80 images that provides a baseline level of model agreement. And then we can find a subset of disagreement sentences, which put models in high disagreement, and alternatively, we can put models in high agreement. And when we visually inspect these stimuli, we don't find any apparent differences between these groups. They both contain similar object categories, and we were confident that they're also natural because we started with natural stimuli. But when we test how well these ANNs can predict brain responses in occipitotemporal regions and visual areas, we find that similar to what we've previously shown in language across high-performing vision models, the neural responses to agreement sentences were predicted best, shown in orange here, followed by random and lowest for disagreement images. And to conclude, we think that disagreement among ANNs leads to divergence from brain representation in OTC. We find that parts of the representation spaces that are shared or universal across models converge with brain representation. And collectively, we think that the similarity between artificial and biological neural networks could arise from universal representation they both learn. Thank you.
Hi, everyone. So I'm going to expand in my talk about this idea of the interanimal transform class, and also a theme that's been coming up over and over today about how strict or flexible our comparison method should be, and hopefully by the end you'll have an answer. I just want to thank my great collaborators first. So we think that there's two key desiderata that you might want from a model-brain comparison method. One is that a good model should be able to make accurate predictions of brain activity like firing rate so that you can actually use it for scientific understanding or engineering applications. But in addition to that, you also want your comparison method to be specific enough to basically tell when a model is more similar versus differentiate between more correct models and less correct models of the brain, or more similar models versus less similar. That requires being able to tell when two things are similar. For example, responses from the same brain area, but also tell the difference between responses from different brain areas, just as a concrete example. But you might worry that these two desiderata are sort of in tension with each other, because imagine if you're just optimizing for productivity, you might think, well, let's just choose as flexible a method as possible, like linear regression, or go all the way and do like a super nonlinear MLP. And on the other hand, if you wanted specificity, you might use something really strict like soft matching or some sort of one-to-one matching method. But this is kind of strange because it almost seems like the goals of engineering and scientific understanding are diverging here. And you might think that the reason you want a good model in the first place is so that you can achieve both. So I'm going to explain now why these goals don't have to be in tension with each other. So here's one failure mode of specificity. If you're like visualizing the distances under some comparison method between response profiles and the different colors are like different brain areas, obviously just mashing everything together and making everything look the same as everything else is not good. That's clearly non-specific. So I think an MLP might do that in some cases, maybe. On the other hand, here's another failure mode. What if everything looks different from everything else? Well, that also seems non-specific. You end up with a mush here as well. But this is a failure mode that hasn't been as widely recognized. And so what you really want to do, and so that's basically saying mere separation between responses of different types is not enough. In order to get specificity, you also need to identify similarity between responses of the same type, like from the same brain area. And it's only then that you end up with this nice, clearly differentiated structure between brain areas when you're, for example, comparing across subjects. Okay, so we quantified this, or made this concrete, like we incorporated this insight into our metrics by using the silhouette score, which has two numbers that keep track of these two dimensions of specificity, which is the same area identifiability and the different area separability. And then we evaluated a bunch of comparison methods on a simulated population of mice with neural networks with different random seeds for different subjects, and we evaluated productivity and specificity using these metrics. And when we did the first thing we wanted to try is what happens with linear regression. And it's really surprising that when you look at the same layer across these model subjects, the linear regression score predictivity is not that high, especially for the intermediate layers. And when you first see this, you might be really worried, like, what if the model subjects aren't even that similar to each other? And what does that say about the animal population? But fortunately, there's an additional result, which is that if you do the linear regression before the activation function at each layer, the similarity scores are really high. And what this is telling us is that if you want high predictivity across the model subjects, you need to somehow take into account the effect of this activation function. You've got these things that are linearly, sort of close to linearly related, but they're being warped on either side by the activation function. You have to take account of that. And one natural way of doing that is to first invert the nonlinearity, then do your linear mapping, and then reapply the linearity on the nonlinearity on the other side to predict the firing rates. This is, of course, challenging if you don't exactly know the nonlinearity, but the point is you have to somehow take into account the details of the biological mechanism in order to get these high predictions across subjects. So we did that on the model population where we actually knew the nonlinearity, and we found that the more precisely we took account of the nonlinearity, the better the predictivity scores, with the best version being the prelinear soft plus, which is the pink bar. 1 minute? Or, 30 seconds, okay. So a good transform must take the biological mechanism into account. Now, here's two contrasts to our biological transform. Here's a super flexible thing. The 7-layer MLP trained on all of ImageNet train, 1 million data samples, does not do substantially better at prediction. On the other hand, soft matching, the really strict one, does much worse at prediction. Now, the key point is, does it do better at specificity because it's much stricter? It actually does worse at specificity. And the reason why is clear on the next slide. Soft matching is making everything look different from each other. And on the other hand, the MLP is making everything look kind of the same. What you really want is this sweet spot, where you can kind of differentiate the different areas and at the same time get high similarity for the same model layer. And so really what we want is the interanimal transform class, the narrowest class of transforms that maps the responses across subjects with high accuracy. That's the sweet spot between strict and flexible. So you want just flexible enough to get this high accuracy, and then stop there, no more flexible.
All right, there we go. All right, I'll dive right in. So when comparing artificial neural network model representations to the brain, one predominant approach is to subdivide the model representations by layer. This enables us to do nice things like evaluate hierarchical correspondence, where early model layers have higher similarity to earlier cortical layers in the sensory processing, and later model layers have higher similarity to later cortical models in sensory processing. But we know that this hierarchical correspondence is not perfect. There are many layers that have higher representational similarity to these different areas. And in performant modern deep learning architectures, they all share this residual style of architecture. So this occurs across all layers in transformer models and all of the stages, or all layers within stages of convolutional network layers. And so what's happening here is that instead of the output of one layer feeding directly into the next layer, they're all outputting and modifying a shared residual stream. And so what's happening is that you have late layers in the model that can still have access to fine-grained, the same image patch embeddings in transformer models. So what can we do? What are other ways that we can subdivide the representations to understand how models relate to brain representations? So one approach here is to look within these activations. So if you look at the activations in transformer networks, they're organized by tokens, which correspond to each of the individual image patches and the dimensionality of the models. And so each of these dimensions spans all the full field of view of the image. And so this work here is just showing how you can do it dimension-wise and compare and relate the models based on these dimensions. So start with centered kernel alignment and ask a simple question of how does the score change when you remove a single model dimension at a time. You can visualize this for all the individual model dimensions and analyze their contribution to the overall representational similarity. I'll say one of the main contributions here is to factorize the computation of this such that computing all of these scores for all the dimensions in the network, say 10,000 of them, is almost as quick as just combine the single score for the similarity score for the network. So what this enables us to do is to compare the similarity score contributions for all the dimensions across a large transformer model network and find the highest contributing dimensions. And so what we see when we compare this across different cortical regions is that the highest contributing dimensions are distributed across many of the model layers. And that if you look at the brain similarity as typically done in the middle here with the best layer, that's higher than if you look at all of the representations across all layers. But if you just take the best, these top with really small subset of model dimensions, you get a really high similarity score. So I'll just say that this is also a tool that can be used to analyze the representations when they're projected or rotated to understand the different representational properties of the encoding in these models, and that this is not the only, applying to CKA is not the only way to do the dimension-wise analysis, but just it can motivate other types of tools and other types of approaches that are looking at this full network layer, or network level dimension-wise analysis. And I'll stop there and thank the collaborators.
Not this one, but the other one. Oh, sorry. Dumb question. How do I? Nice. Thanks. OK, so things in the image reconstruction world have gotten really good. This is an image shown to a subject in the fMRI as part of the natural scenes data set on this side, and there's the same image reconstructed from the fMRI. It's from a 2013 or 2023 paper, but there are many others, and there have been many since. And things just keep getting better and better. Like, according to image similarity metrics, we're getting closer and closer to the ground truth images. But the question remains, are we really getting better at extracting signal from the brain, or are we just getting better at image generation? Like, you can imagine the case where the model just knows what a plane looks like more than it used to. And coincidentally, the images started getting better as soon as the diffusion models came out. So we need a metric to distinguish between these cases. So we propose brain bits, which is how much of the brain signal do you actually need to get reconstructions that are just as good. So we insert an information bottleneck into the reconstruction pipeline, and we restrict this bottleneck and see how much the reconstruction quality degrades. Basically, so we do this with a couple existing approaches. We find that we can constrict this bottleneck quite a bit. With information 1% the size of the original input, we're able to get images that by eyeball look just as good. Here's one image, here are a couple more. And not only that, just according to the similarity metrics used in the prior work, these images are just as good. And the takeaway message is, the metric is going to determine where progress is made, and if we care about studying visual processing in the brain, we need to actually look at how much signal is extracted from the brain, not just the image quality. Thank you. OK. Cool. So today I'm going to talk about some, pilot work. This is just some work we've started on a new kind of way to answer a question that we all care about, which is how do we represent tree structure in the brain? So generally, when we think about geometric representation in the brain, we can think about either taking in some stimuli and then either representing it as some sort of tree structure in some sort of hierarchical way or some sort of graph structure below. Right? And this is, different from a lot of the many prior studies that look at how, order of processing or hierarchical processing. This is asking a very geometric question instead of, how am I taking in the information and then putting it into activity or some kind of representation in the brain. And so the way that we're thinking about this is through hyperbolic spaces. So hyperbolic spaces are something, have very nice properties about capturing hierarchical structure. So particularly at low dimensions, you can actually capture an entire tree. And there's a lot of proofs that go in the literature about how you can build different spaces that do this. So this is a Poincare disk. This is in 2 dimensions. It's actually able to fit an entire tree in 2 dimensions. And this is really nice because we have now started building networks that are actually able to take in word representations and encode them in hyperbolic space. So we can start to answer questions about how in the brain this kind of information is also encoded, instead of just answering questions about where in the brain it happens or the kind of computations that are undertaken that are most similar between networks in the brain. So as sort of a pilot study, we started looking at hyperbolic embeddings and used them to reconstruct WordNet. You can train a hyperbolic network. You either use a Euclidean network or a hyperbolic network and have different dimensionality for your representations from 5 to 200. You can see that at very low dimensions, we can actually look at the reconstruction reconstruction performance here, where lower is better for the top row and then higher is better for the bottom row. For both of these kinds of ranks, we're actually able to get way better reconstruction for the Poincare embeddings at low dimensions. And then at high dimensions, the Euclidean embeddings do better. And so what we're actually able to do with the hyperbolic networks is we're able to get some kind of, filter based on dimensionality between low dimensional capturing all of the tree information and high dimensional capturing a lot of the graph information. And so what we're proposing here is some kind of hyperbolic representational similarity analysis, where we keep all of the same structure of representational similarity, but use hyperbolic distances. And the nice thing here is, as a metric, this is answering a rather orthogonal question that we weren't able to answer before by considering a new kind of network design. And so I guess as sort of a pilot piece of information, this is not, we have not done anything comparing to the brain. We can just compare, I guess, hyperbolic information and try to see whether like RSA is able to tell you anything about similarity between graph structures. In general, hyperbolic embeddings have way higher RSA and are able to capture graph information way better. And I will just leave it there. Cool. Thank you.
All right. So we want to measure representational similarity. The problem is that there are many ways to do that. And if you look closer, there are even more ways to do it, because each of these methods has its own set of hyperparameters that you have to choose. So if you actually take that into account, the picture is more something like this, where you really have a lot of measures. And so the question is, I don't know, what do we do with that? How do we choose which one we want to use? Maybe we are lucky. Maybe most of these metrics are equivalent. Or maybe there is one over there that's, I don't know, better than all the others. And if we find it, then we can just discard all the rest. So to try to address these questions, we have developed a new method to compare metrics by leveraging the differentiability. So the method consists in taking one reference data set, X, here, and another randomly initialized data set, Y. And what we do is to directly optimize Y to maximize the score with X by directly differentiating through the score function. So we can visualize what it looks like, for example, for this linear regression metric. As you can see, it starts random, and then it gets more and more similar to the reference data. We can also quantify this by looking at how well each principal component of the reference data is captured during the optimization. And if we do that with other metrics, we found that you can have many different results. Like for example, for this CKA metric, we found that the PC2, so the second principal component of the data, is captured for much higher scores than for linear regression or other metrics like Procestis Angular. And so this kind of suggests that maybe there are some differences between these metrics that we should care about. So what about real neural data? So we analyzed data from the Siegel 2015 data set. And what we did here is to try to decode the different task variables from the optimized data set directly. So we start on the left, and as the score gets better, we found that the decoding accuracy also increases and approaches the level that we can get from the neural data. If we do that for the other metrics, we found very different things. For example, if you look at the score of 0.9, which sounds like a good score when the max is 1. For linear regression, we found that not all the variables can be decoded as well as in the neural data. For CK, it looks even worse. And for Angular Procestis, it looks quite good. So maybe there is maybe one measure that's better than all the others. Maybe if we have a metric A that's better than all the others, would mean that for any metric B, if A is high, then B is also high. We could also have other things, like maybe they are just equivalent, or maybe they are completely independent. So we tried doing that with our metrics. And for that, we took one tuple of scores for each of these metrics, and we used that as the optimization target for our method. And then we ask if, starting from a randomly initialized data set, if we can reach the targets by optimizing the randomly initialized data sets. And we also look at when the optimization gets stuck, because maybe there's some of the scores that are not reachable. So we did that for many optimization targets, and we get something like this, where here we found that when angular prosthesis is high, like 0.9, it always implies that CK is high, but not the other way around. And we found a similar relationship with linear regression, but interestingly is when we regularized and also cross-validate linear regression, we don't get that. So it seems that the picture here is a bit more complex. So we found that not really all metrics are equivalent. We can find really high differences between these. And also, some high scores imply others, but it's not always the case. And so if we come back to this picture, we really need evaluation methods that are scalable to try to navigate this complex landscape, which is also why we have worked on a Python package to try to register all the metrics that people have published. We currently have 83 metrics from 14 papers, and we'll keep adding them. And we also try to provide a unified interface so that you can easily use any metric from any paper. And with that, I would like to really thank my collaborators, and here are some of the links if you want more information. Thank you.
Okay, so thanks so much for bearing with us. We hope this was informative. I now realize in retrospect that I had a 3-hour aspiration for an hour-and-a-half time period. But the general intent here was to introduce you to the broad variety of things that are happening in metric space, the very exciting things, and to get conversations going that we hope will persist into the future. On that note, I'll end with some final, pardon? Oh.
Try the other side.
Other side?
This thing, the other side.
This thing, the other side. I see. Oh, I see. Wait. It's plugged in. I think, oh. Gosh darn it. Okay. If not, we'll do it live.
There you go.
Okay. So I just wanted to say a final few things. No, this is the wrong set of slides. That was the original amount of slides that I was supposed to present, but then had to cut. Here it is. No, it's not this. Where is the right one? Oh, I see. It's right over here. I have to look on this screen down here. Boom. Okay. So few final closing words. All right, there we go. Okay, that's good enough. So a few final closing words. First off, even though there were many people here and you got a broad sampling, there were actually quite a few people that contributed abstract and/or code and/or other works about metrics, and I just wanted to highlight a few of these wonderful people here. I encourage you all to go and check out their work. They've done some phenomenal thinking in metric space and also a lot of good experimentation, and it would be worth your time to go and check that out. So the next thing, I basically tried to just write down all of the hot takes that I was going to give at the end of this session in this slide, but the overarching view that I wanted to propose is somewhat predicated on this idea that the best thing that we can do, given the broad diversity of metrics in metric space, is to figure out how we can do strong science at scale. What does this mean? It means a few things. It means, for example, that we should never replace data with theory. Mathematical claims about whether or not a system has a certain competence must be tested. We should do this at scale. We should not necessarily make commitments to any principle that hasn't been empirically validated. And what does this mean? It means we should simultaneously incentivize the engineering of scientific infrastructure while not mistaking engineering for science. This is thing like, not confusing prediction with understanding, and also breaking that down a bit further. There's this idea, I think, that engineering is somewhat powered by coverage. More data, more samples, get all the information that you can, and then model it. But science is somewhat powered more by this idea of parsimony. We want to be able to articulate in a limited amount of information something that then conveys a lot of structure that is useful to us in the world. And so this brings us to some of the questions that people had. And these were basically the themes that GPT and I summarized. There were not a lot of responses, so GPT was a little confused as to what was going on. But broadly, we can break down some of the responses into 2, 3, 4 themes. The first is that there's actually maybe even more uncertainty here about what we're talking about than even just the idea of a metric. Representation, computation. What do these things mean? What's the difference? How do we define them? Should we define them, or should we allow them to be flexible concepts? The next thing, are models learning the world or a reflection of the world? Is there a difference between these things? Are claims about the difference in the scale of data consumption between models and humans meaningful? Are, for example, the vision and language models that are converging in the Platonic representation hypothesis learning something that's a correct structure of the world? Or is the internet dominated by us taking pictures of things that we talk about and talking about things that we take pictures of? Finally, there's a direct quote here that I love because this is actually a theme that has come up many times. There's not much of a battle here, so let me ask this. Why should we choose any of the presented metrics over others? It seems like every presenter has their own preferences for what a metric can capture. Why is any of these preferences more correct than another? And the answer is, they're not. You have to go out and test these things, run the test yourself, figure out the science that you believe in that makes it clear to you what kinds of hypotheses you need to set up, what things you need to control, what confounds you need to take account of, and then do it. Now, how do you do that? Well, we're only one person at a time, so one of the best ways is to lean into people. What do we want from our model-to-brain mapping metrics, and how do we get it? Well, we want many different things, and we have many different ideas about how to get them. But let's do science together to figure it out. This partially means moving away from commitments to certain kinds of infrastructure or pipelines or dedicated procedures, and allowing us to kind of use tools, i.e. BrainScore and other types of platforms, but in a way that's far more flexible and allows people to interject the kinds of opinions that they have into their ability to do science with these large-scale benchmarking platforms. And with that, I will conclude and say thank you very much, and I hope many others of the team will be happy to speak with you after. Oh, last thing I forgot to mention. In service of this, there actually was a whole bunch of data that was collected specifically for Battle of the Metrics in what many of you saw online as the empirical sweep. We will be hopefully pushing this data out soon. It needs to go under some scrutiny based on some of the metrics that I and GPT ran last minute and make sure those are correct. But in service of offering data to the community, we'll be publishing things. This is not a leaderboard. This is a data frame. You can go in and aggregate it and look for the factors that are interesting to you or not.